Hello, welcome back to our channel. To listen today, we are talking about ATAC-seq again. So last time we were talking about ATAC-seq. I would like to kind of approach this topic again because there are many questions still to be answered. How to analyze ATAC-seq data. Okay, so what will happen today? So I will first talk about ATAC-seq, how we're going to analyze it, what are the approaches we have in our platform, in our set of tools which we have here. So first of all, let me start from a little presentation just to let you know what we are talking about. I will share the screen. So we again we'll start from ATAC-seq, what it is, and well, there is a pretty good overview of ATAC-seq data analysis methods in this review, which is already 2 years old, but it's 3 years old, I must say, but it's pretty good. It accumulates a lot of knowledge about the methods which people are using to analyze ATAC-seq data. So ATAC-seq is very popular now. If you look at the number of publications, basically they are growing in the last years at ATAC-seq, and that's not a surprise because that's very kind of flexible and quite universal method to reveal the regions of DNA which are opened and ready to accept regulatory machinery and open chromatin, and they are going to regulate the genes. And that's a typical picture which you get from ATAC-seq, and that's a picture you got from this paper. So which actually accumulates more or less those steps which you usually have. So first, ATAC-seq is done by special enzymes, transposase TN5, which cuts regions of DNA when they are not covered by anything. First of all, by nucleosomes. So if nucleosomes are shifted around, so there is a window of sort of open DNA, which is now accessible for this enzyme, and start cutting it. And then we got these, fragments. Then we are sequencing them in an NGS sequencer. And then we map those fragments back to a genome. And with that, we see we call them peaks. We see those regions which are open. Sometimes, actually, some of the regions are occupied by some other proteins, for instance, by transcription factors. And that's a topic we are going to talk today. And then those areas may be, again, protected from this enzyme to cut. And sometimes you see that in the profile of ChIP-seq data, that there are some little valleys where you can expect there are transcription factors sitting. But basically, the general concept is the whole area is more or less open for regulation. And first of all, the question is, if there is a transcription factors which are regulated activity of my gene, so they might be binding to the areas of chromatin which is open. So that's exact places where we should search for the motifs for transcription factor binding. And there is a number of methods which are available now on the in the internet or in different packages, starting from the very beginning. Once you have your sequences, you have to map them, you have to check the quality of them, of the mapping, and then there's a number of tools which you then apply afterwards to the areas which are defined. First, you have to find those motifs, those peaks. There are a number of peak calling algorithms, and we were talking about that last time. But then what we are talking today is more or less the last part, when you already know your peaks and you want to use these regions to interpret your data. Basically, you have to understand how these regions are regulated, which transcription factor might be binding to these regions. And then actually from that, you figure out what happens with your system. So we will talk about, basically motif enrichment in those areas, in those peaks. And then the next step from the motifs, we will talk about actually pathways and networks. And that's what I'm showing here in this slide. again and again, and I will show it quickly today, that actually what we are talking about is sort of what happens in the cell. So, and there are areas of genome which are regulating the gene expression, like it is shown here, and that area is detected by methods like ATAC-seq, and then where we actually try to find the motifs for the binding sites of transcription factors. And then once we found them, from that we can assume which factors, maybe which transcription factors, acting still in the nucleus of the cell, they can bind to those places and regulate the genes. And then we go more upstream. We're calling it upstream analysis. We are trying to ask a question: are there any master regulators which might regulate activity of all these transcription factors altogether? And once we found those master regulators, they might be a very good potential drug targets. If we are talking about some pathologies we are analyzing. And okay, so we are using different databases which we have in our hands. So here for the motif search, we use TRANSFAC, the collection of motifs. For the pathways and the network, we use TransPath. It's another database from us, which collect the network of signal transduction interactions, which we collect manually from literature. And then finally, a database called Human PSD, which is a database about drugs, about biomarkers of different diseases and so forth. So, and then all this is done with the help of various types of omics, expression with transcriptomics. The network can be facilitated when you have proteomics. And what we talk today is about epigenomics. So ATAC-seq is one method of the whole area of experimental methods to study epigenomics. So what happens not in genome, but above the genome, on epi level. All right, and in this paper, there was a very good presentation of various algorithms which we have to analyze ATAC-seq data. I mean, these are available in different packages. In our platform, we have few of those algorithms which we are mainly using, like for instance, MAX2 to call for the peaks, or other types of algorithms to analyze actually the form of the peaks and things like that. But okay, so just to show how it works, let's start right away from a particular experiment from particular data. And today we are talking about a tumor called Desmoplastic small round cell tumor. It's a pediatric and very aggressive, but rather rare cancer, which is happened in the abdomen. And the very bad thing of it, it's actually very difficult to cure. So it's random. It's also rare. It happens quite rare in the population, but once it happens with children, it happens quite some time, and then there is very few means to battle it. So, and the very important actually part of that tumor is that it's characterized by a sort of big cells which have a special form. That's why they call it a small round cell tumor. So those round cells, they got so they actually are very resistant to very many different therapies. And today we are going to use this data from Gene Expression Omnibus. These are screenshots from that paper, or from that set of data which just published in December this year. So the paper is not published yet, but the data is already free, available. It's from USA, from New Orleans. Oh, actually, I've been in New Orleans. It's a very nice city. Okay, here what we have is actually a collection of data. First, and here in this entry on Gene Expression Omnibus, we can get ATAC-seq data directly, and we'll see how to get it. And also, there's actually gene expression data from the same system. And what they have done in this experiment, they actually have two cell cultures. One is a cell culture of those spheric cells. So those small round cells, which are very bad in treating. So, they compare it with other type of cells, which have a different form. They are adherent, so they are kind of flat, and they are more easy to treat with different chemotherapies. So they are dying. Although this round shape, they are not dying. With those therapies which are available, or they are actually quickly develop resistance towards those therapies. And how to get the data? So actually, let me just go online. Let's go online to our GeneXplain platform to see that data, and then we can have a look in the gene expression omnibus to see the origin of that data. So for that, we log in into our system with a password which I have. So if you have license to our system, you have your password. So with that, you get you get directly to the search engine when you can search for all kind of information, for instance, in TRANSFAC or in TransPass or human PSD, or you can go directly to GeneXplain platform. And actually today, I'm going to first show to you actually a new tool. Well, it's already existing for quite some time, but I haven't talked about that extensively during my coffee breaks. It's called Genome Enhancer. So Genome Enhancer, it's a tool to analyze multi-omics data. And what it's doing, it's really combining all types of data. For instance, in this case, we have ATAC-seq data, and we will have also gene expression data. So you'll see what kind of data we have just in a moment. But then what happens, you just put this data in this tool, and it will know how to combine epigenomic data, and gene expression data. If you put there transcriptomics and proteomics, it will know how to combine it. If you put it, all kind of other types of data, so we'll talk about that maybe in future. But today our topic is really epigenomic data, ATAC-seq data. So the data which we want to see today is this one. I have to search for this gene expression omnibus entry. All right, so here we are. So that's the data we wanted to analyze. And as I said, actually, there is a set of data. So first of all, this ATAC-seq data are available as various types of format. But if you want to really grab the raw data, which means the FASTQ files, which were done in that experiments, you go to SRA run selector. And here, what you need is just to, I showed that last time, you need this accession number from SRA entry, and then you put it into our platform, and you will download this data. So what we did last time. So actually, we download these FASTQ files, we align them with with various tools. Actually, we align it to the genome, and then we run the peak calling algorithm, which was MAX2, and we found the peaks. So I will not do it today. Actually, I will just show you that, okay, we have this SRA data. So what you have to grab these accession numbers and put them into our platform, and then you will get this data. So already converted to FASTQ files. But once we did that all, so actually what we got at the end, we got the peaks in BED format. And I want to show you how to upload this data into the GeneXplain platform, and especially into Genome Enhancer. So you see it's already uploaded, but I want to simply show you how to do it. It's very easy. You can either add this file from your folder. So I have these files on the desktop. I know it was in download. And these are the peaks in BED format. So you simply click open, it's imported. So you click import, and then it recognizes this BD file. You have to tell what kind of build it is. So in this case, it was HG3038, which is the last build. And then you specify that's epigenomics, because it could be transcriptomics or all other kind of data. So in case of BD file, most probably it's epigenomics, and the guess is correct. And then you import it, and then you have it inside the platform. So now it's ready, but as you can see, we got all other types of data. Especially we will need also transcriptomics data. And transcriptomics data here is present as already pre-processed data when the authors of this work, they actually did RNA-seq. They pre-processed those data with PIC. Well, actually, they again aligned RNA-seq on genome. They basically calculate the counts for each gene, and then they compared the genes which are expressed in the level of expression of the genes in the spheric cells to compare them to the flat cells. And then they got this actually number of genes, it's around 2,000 of genes, which are significantly differentially expressed. So which means they have this calculated this log-fold change, which is, and then p-value and adjusted p-value, and these are those genes which have either significant p-value, so it's not all significant, so we can upload first this set of data, and then we can filter it. So that's all gene expression data, but we still have to filter which of them are up-regulated and which of them are down-regulated. All right, so we have this file. So what we do again, we select this file from our folder, and now it is a different types of files. So it knows, okay, that's a tabular file. So it has those number of columns. The first column is ID, and it's ensemble, and it's definitely a transcriptomics. So it's gene expression. So you simply import. And now you have transcriptomics data and ATAC-seq data from the same experiment. All right. So then you simply click next. And that's exactly, I did already before, but let me show you how to do it again. So now we uploaded these two types of pics, right, and the expression data. So you simply have to, so you see here, these are the pics, PIC files, and this is the expression data file. So basically what you do, you have to drag and drop them here in this gray box. I already did. And then when you have them here in the gray box, you click next. And now you can specify what types of data you have. So, and here we have spheric cells, and we have adherent cells, so the flat cells. You can either, what I did actually first, I have the spheric and adherent, and I have put the peaks which were found in the spheric cells here, and the peaks which were found in adherent cells here. And then I have put here the log-fold change for gene expression in the spheric cells, because it's already comparison of spheric cells to the adherent cells. And that was one type of analysis. Another type of analysis, actually, I did compare these peaks found by the spheric cells to the adherent cells, and I found the difference. That way, how to find the difference between two sets of peaks I showed last time. So I have another set of peaks which is a different peaks, and now I prepared this spheric filtered category which have gene expression and the difference between these two sets. So basically, the same you can do in other category. For instance, if you want to compare a gene expression of spheric versus adherent cells, you drag and drop this log-fold change here. And for instance, you want to compare not just to the different peaks, but to real peaks which were found in this adherent in the spheric cells, right? So you just drop here and drag and drop them. I have to here. Probably it's because it's the same entry before. So okay, so you can have just one set of peaks or two of them. If you have two sets of peaks from two individual experiments, they will be combined together. If you have just one set of peaks, you just drag and drop here and you have just one set. So you have your new category, you call it, and you can give a different name to this category, exactly what you like. So once you have those categories done, so you go next. And then what you would do here is you would select the category you want to analyze. So, in fact, we would like to analyze here this new category, for instance, and it's just one category. You don't need to compare to anything else. So you just remove this second category. In fact, you can put here more categories, like 3 more or 4. And then in this case, it will be iteratively comparing these categories to each other, or it will do the clustering of them. So there's various ways of analyzing this data. But in this case, we will just use one category. And what we'll do is actually to select the disease. And actually, it's a sarcoma type of tumor. So we can select this sarcoma, for instance. So this disease selection will help us to, at the end, once we find the master regulators, actually to find which particular drugs can be used against those master regulators. So once you're ready, you start the analysis, and it will take a few hours to generate a report. So I will not run it right away because I already did it. So I just go directly to the analysis reports. And that's the one which we analyzed. This here filtered. So this is a category which includes transcriptomics data and the difference, the set of ATAC-seq peaks which are different between spheric cells and adherent cells. And here it generates two types of report. One is called brief tail. It's a very brief report, just to have a look at it, or the full story, which tells you the whole story, what it actually found. And that's let's go through such a report, and then actually we can go back to the whole platform to see what type of data do we have there, how we represent them, and what else we can do with this data. But let's first have this report. So what this report is telling us is actually first analyze this data, select upregulated genes out of this whole set of genes which we uploaded, and it runs analysis of motifs in the peaks, in ATAC-seq peaks, which are first of all different between round cells to the adherent cells, and second, they're actually located in vicinity of upregulated genes and downregulated genes separately. So it's not anywhere in genome, but actually what we do, what system is doing, it first selects these upregulated genes, then it takes area around it, like 5' region, 3' region of the gene, intron regions, and then it looks whether those peaks which you have is located somewhere nearby. And once it found them, it uses this motif enrichment algorithm, which we were talking before, not just in the promoter of those genes, but in the peaks which are located in the 5' regions of those genes. In case if the, and we'll see it a bit later, in case if the upregulated gene has no peaks around, in such a case, the system will take its promoter to that set of to the set of regions which we is going to analyze. So it's a mixture of peaks and the promoters of the upregulated genes. All right. So and then it reports which transcription factor motifs are enriched in those regions, and selects these 3, for instance, as interesting, most important factors. and then it goes upstream and find master regulators. Let's have a look exactly what it's doing. So so that's the set of data we were analyzing, expression data and ATAC-seq data. And then, as I said, first it selects those genes which are upregulated. So you see here the table of these 10 top upregulated genes. If you click here, you see the full table. Let me see. I have to go back actually. Let me go back to the report. All right, so and then I see these genes, and then what it's doing, and I was talking about before, actually it takes those genes simply still without without ATAC-seq information, just expression, gene expression, and start mapping them on gene ontology terms, just to have a an idea what kind of functionality it's changing in these spheric cells compared to the adjacent cell. By the way, why we actually compare it? Because again, coming back to the very beginning, so those adherent cells, they are actually sensitive to many different types of therapies, although the round cells not. So if we compare these two conditions, maybe we will know what is the difference, and then maybe we find the mechanism which maintain this difference, and then we can find vulnerabilities or master regulators inside that mechanism, which can lead us to idea what kind of drugs we can use. So, okay, so as you can see, these genes, which are upregulated in round cells, they have interesting geoterms like regulation of cell migration, mesenchymal cell development, and that's very important, actually, geoterm. We are going to talk about that later. Then what it's doing, it's taking those genes and start mapping them on signal transduction pathways, canonical ones. So we know in many tumors, the pathways actually get different. They are rewired, and we were talking about that many times. But still, if you some parts of those rewired network are actually canonical pathways, like kinase cascade, which is very like backbone of many of these networks. And then there are other pathways which are different here. And you see among the upregulated genes, many of the pathways which belong to actually metabolism. So a lot of metabolic pathways actually changed in these round cells. And then when we map those genes on different type of diseases, or biomarkers of different diseases, strangely enough, we of course see those genes which are characteristic of neoplasm, but neoplasm, for instance, of nerve tissues. And in general, like glioma, also cardiovascular diseases. So it's very interesting set of genes, which is not very typical for like tumor. Or if you have just genes which belong to rapid proliferation of cells, you immediately see here a lot of different types of tumors. But in this case, these cells are quite different. The same we do for the downregulated genes. And here you see a very clear picture. Cell differentiation, cell development is downregulated. So these cells are not going to differentiate. They're going to stay undifferentiated. And that's a very important characteristic of those cells. And again, when we map them on pathways, you see these special interesting pathways like TGF-beta pathway, pathway of ERBB1, so of the EGF cascade, and other interesting pathways. And as for the diseases, again, we have kind of mixture. But it's like a teaser to see what these genes are doing. But then what we really want to see is actually how those genes are regulated by the transcription factors, especially those binding sites are located in the ATAC-seq region. And that's exactly what happens here. So here what we do is, it is described here, we call them enhancer regions. So actually those regions with open chromatin, which are shown by ATAC-seq on one side, but also actually which are characterized by a set of motifs which are capable of regulating genes in up or down regulated way. So we call them enhancers in this terminology. And this what happens. We take 5' regions and actually the whole genomic regions around the genes, and we search for the peaks. And then when we found those peaks, we start looking for enrichment of those peaks by motifs, and not just single motifs, but motifs in combinations. And it found this combination. This is actually, we call them modules. So it gives you idea of, this is actually how the transcription factor motifs are called in TRANSFAC. So these are so-called these are names of positional weight matrices, where you have the name of the transcription factor family, like FOXP3, for instance, or EGR family, or TCF12. And then you have V dollar, which means this is a positional weight matrix for vertebrates, which means you can use it to analyze human data, mouse, rat, or any other vertebrate genome data. And then there is other information, whether a certain number or whether it's based on quality 3 sites. So that's the whole story, how we build those positional weight matrices and put them in TRANSFAC. And then those matrices, we have actually in TRANSFAC around 7,000 positional weight matrices for different transcription factors. So it first applied all of these motifs to sort them in these regions, in the peaks. And then it started it's actually applying genetic algorithm to find a combination of motifs which are located in a small window. Like in this case, combination of these motifs is located in a window of a size of 143 in average. So this is a small window inside these peaks, which are inside 5' regions of the genes, where those motifs of these 5 different transcription factors are together. And that's another module, module 2, which have another set of factors which are also in a similar window located in those peaks. And already here you see some interesting factor which tells you some story. For instance, so-called SOX factors, SOX5 and SOX2 and different SOX factors, they're actually playing an important role in dedifferentiation. So these are famous transcription factors which are used to de-differentiate your cells, to create pluripotency of the cells. And this is not the only factor, but you can start seeing them in these motifs. And this histogram show it's actually, I would say this data is not very clear. It's pretty noisy, but still what you see here, this red bars shows the concentration of those factors, binding sites in the peaks, which we consider, and the blue bars are actually concentration of the same sites in the other random regions of genome. So these two sets of the bars are differentiated significantly, although they're quite overlapping already. So it's telling that the information is pretty noisy, but still this signal can be detected in it. Right. Let us have a look actually at this table, how those peaks are actually, how those sites in the peaks are seen in the first place. So let me sort by this score, and then you see it more clearly. So this is, for instance, the first peak, which is located in this gene, CCD C13-7P. And actually, it has a very high CMA score. And those little, dots or arrows here they are showing this binding sites for this module of transcription factors, which are clusterized together, and they are located here inside this quite a big peak, but in a near this, well, actually at the left side of the center of the peak, you see this cluster, and here you see those sites which are found in this cluster. So in another gene, this one, you see another cluster on the right side of the peak. You can see those peaks also on Genome browser, and I think we will see that a bit later today. All right, so this way we see how those found enriched motifs are found in the peaks. But we are going further. Actually, the further we want to look at the downregulated genes, and in the downregulated genes, we again see a set of other factors. We see actually again SOX factors, and we starting to see other factors like SMART. SMART are another factor which plays a little bit of role in pluripotentity as well, but you see also other factors here. We'll see a bit more in an open situation when we open this data in the geneXplain platform, but now it's just a summary of finding. So now we actually know a bit which transcription factors are involved in regulating of my genes. But as I said, we want to go further. First, we can list those transcription factors. We can map the frequency of binding sites of this for these factors in the peaks which we are studying compared to the other random regions of genome. And finally, we go to search for master regulators, and that's a typical picture or diagram, so we can open it a bit wider of such results. So what we see here at the bottom, we see those genes which are upregulated in our experiment. So basically, these are genes which are upregulated in spheric cell compared to adherent cell. There above them, we see those modules, the composite modules, so the collection of transcription factor motifs, positional weight matrices, which were found in the peaks located in the promoters of those genes. And then we see those transcription factors which are associated with these motifs. You see this CBP, SOX5, FOXO2, so O1, actually those which we mentioned, EGR1 actually also expressed upregulated by itself. The gene encoding EGR1 is actually upregulated here. And then the next step is searching for master regulators, which means we're going into our network of interactions from TransPass, and we start going upstream to find which of the proteins in the system, which of the signal transduction proteins like kinases, receptors, ligands are connected to those transcription factors on one side, and also they are encoded by the genes which are regulated by these factors. So for instance, this gene VEGFR2, this protein is encoded by the gene KDR. And for instance, RBB3 factor, RBB3 protein is encoded by RBB3 gene. So you see these are so-called positive feedback loops, which means this protein activate the set of transcription factors which are through these open chromatin regions. They may activate the gene which encodes the same protein. So it's like autocatalytic loops. And exactly these kind of loops are searched in the Genome Enhancer. And these are best supporting evidences to tell that these are master regulators. So this is a set of proteins which are not only capable of activating those transcription factors, but they also activate the genes which encode themselves. So they really getting into autocatalytic loops. And among them, you see, of course, such factors like EGF receptor, RBB3. You see also factors which are well known, like VEGFR receptor, which are very well known in many tumors. You see many kinases involved in tumors here, but very specific set of kinases, which is actually characteristic for these spheric cells when you compare them to the adherent cells. All right, so we see this picture, this diagram. Moreover, we actually see the whole list of master regulators, which are found here in this experiment on diagram. We only see just the top set of them, but in general, we can open this as a table. We actually have found more than 207 proteins and protein complexes which can play a role here as a master regulators. And interestingly, if you look at the set, in that paper, which was done by these authors, they found one kinase, which is, I think it was LCK or a different kinase. So one of the kinases which was found here, not at the top though, but it's among the predicted master regulators. So in a sense, we confirmed what was found in that paper when people were analyzing it with very thorough tools here. It's automatically, you just run it, click one button, and you get this set of master regulators. But we are going a bit further, and actually the same diagram, a similar diagram, is shown for the downregulated genes, where you can have actually also positive feedback loops, but in a negative way. Well, maybe we can call it a negative feedback loop in effect, because what happens, these genes are downregulated, and they are controlled. The downregulation is controlled by a set of transcription factors, which kind of playing a repressor role. And this to start them repressing, they have to be activated, or, well, in most cases, it's not the active repression, but remove of activation when you see less of those factors activating your gene. So then the expression of those genes going down, and then you see those master regulators found here for the down regulation. All right, so now we take those master regulators and we go through known drugs which can be affecting those master regulators, and we select those of them which actually druggable, so which are known for various drugs, and we call them potential drug targets. And then basically what happens, we look at those drug targets, and then we look in our human PSD database, and it's actually checking what kind of drugs can be used against those drug targets. And as you can see, in case of sarcoma, we have this drug here, pazopanib, which is targeting this set of targets, different types of kinases, and this is a set which is we found here as master regulators. By the way, among them, this BLK, this was a kinase which was shown also in that paper, so we found it here as well, and it's found here as a target for this pazopanib drug. And by the way, actually, this drug is one of the few drugs which is actually used not only against sarcoma, but against this particular type of sarcoma, these round-shaped cells. So when you Google it, you find those clinical trials with this drug, which is going on right now. So you see the system found this drug automatically just based on a set of upregulated genes plus the peaks from ATAC-seq experiment. All right, so that's like you see, I didn't show much of the tools so far, just showing you the result of the automatic tool, big automatic pipeline. In the rest of the time, let me quickly go directly to the platform. I switched to the platform to show a bit how those data are can be seen inside the platform. So now you get here additional toolbox. So like you can open this type of panels. And this, if you go home, you see the whole set of workflows which are available for users to work with different types of data. But what we are talking today is about these results of this one big pipeline, which is running by Genome Enhancer. And you see this is a result of that pipeline is this particular folder. We just opened this report, so which was here, this report file. But we can open this whole set of files which were generated by this big pipeline and can look inside what happens in each of the step. For instance, these regulatory regions which were created by this pipeline, which is a region, which is a set which overlaps of the promoters of upregulated genes with ATAC-seq peaks. So we can have a look at those regions. For instance, in a genome browser. For that, we open this genome browser here, which also shows us the genes, and then we simply drag and drop here these regions. And if we can actually look at those regions. For instance, this is a region which is found in the promoter. So let me see, where was it? Yeah. So for instance, this is a RAD-Y1 gene, which is upregulated, and this region which was analyzed is in the promoter of that gene. It's just a promoter of the gene. But what about these peaks? So to see the peaks, we can see them. We can drag and drop this these peaks from. Let me see where they are. So we can have all the peaks. So we have these peaks, right? So we can put them. Let's put from spheric. All right, there's two type of peaks. So we can see those peaks. You see the peaks, and yeah, for instance, in this case, this is a region which was selected, and that's it's actually near. So because in fact the bigger regions was analyzed, it's near the peaks which were found in the promoter of that gene. So that's another case. This which the region which was selected to analyze this in this gene which is upregulated, and that actually coincides with the one of the peak from ATAC-seq experiment. And let's consider this, for instance. And if we now put here all the sites which were found in those peaks, so actually from this module, we take these all sites. Yes, here they are. So you see what happens. And actually, that's a nice picture also showing a little bit of this valley. So we have this center of the peak. In fact, that's the center of the peak. So actually, we can also put here a bit more information from if you put here the BAM file, yes. Well, it's maybe not the best BAM file. We have other, not here. All right, so I have them in a different place. So it's these are the reads which were found in that region by ATAC-seq experiment. It's not very high peak though, so you can see, but still the peak calling algorithm considered this as a kind of wide peak. And the center of that peak is somewhere here. So, and then we choose this region to analyze for the motifs, right? So, and then actually exactly in the center of the peak, there are not so many motifs. So maybe that's exactly what happens, when there is a empty place which is not occupied by nucleosomes, but occupied by these transcription factors. They also protecting a little bit this region from this enzyme, and that's why the center of the peak is usually in a little bit of valley of presence of motifs sometimes. So, and you see those motifs for the factors we were talking about. This is one way of finding factors by looking to the combinations of them. You can also look very simplistic, basically just taking each of the 7,000 motifs from TRANSFAC and round them one after another to try to see which ones are actually overrepresented in those peaks. And that is done by site search algorithm. Let me show you one of that results. So actually here we see those overrepresented motifs, right? I think it's this one. It's already converted. Let me see, site table. That's from transcription factors, optimized summary. All right, this is the overall set of motifs which were found out of 7,000 motifs, which were found of differentially overrepresented in those peaks. And it found actually quite many, 1,200 different motifs, which have a p-value of above 0.1. Actually, you can sort by p-value. For instance, this motif for PO5F1 is actually oct factor, is definitely very significantly overrepresented in those peaks. And there is another one, famous one called non-oct, and that's also overrepresented and very much significantly. So even single motifs are actually overrepresented here, not necessarily in a combination. And these motifs tells us interesting story, because these are factors like oct and non-oct and SOX, which we were talking today. These are typical factors for reprogramming of the cells. So these tumor cells are not, they're very special. They are reprogrammed to be very much pluripotent. So it is so-called maybe tumor stem cells. Some people call them like that. And they're very difficult to kill. Still, when you analyze it with such a tool, you can find a set of targets which can be targeted already by existing drugs, and but actually also it shows some new new possibilities for other drugs, which you see also in that report. All right, so that's something I wanted to tell you today. There is one little thing which I wanted to show you as well, but let me first check if there's some questions. So if you have any questions, just type it down. Let me wait a little bit. But I would like to comment on one question we just received recently from one customer. They ask if for the ATAC-seq data, we also use the quantitative information. So the quantitative information is meaning that in this ATAC-seq data, when you have it, you like I showed in the first slide, you not only have the regions by themselves, which we're just analyzing. So we took these peaks and we were analyzing the regions independently what is the size of the peak. But obviously those peaks are of different size. And that's you can see here, for instance, on this picture from typical ChIP-seq, the same as ATAC-seq experiments. When there are areas with small peaks and there are areas with very high peaks. And that's what you see also in our genome browser when you have those, when you zoom it out, you see those peaks more clearly. Well, there are areas with the small peaks and there are areas with the higher peaks like these ones. So, and whether there's possibility to use this information about the height of those peaks, and this is very valuable information because the height tells us actually how often that this place is open for accessible for the enzyme. It means the more accessible it is, the more active it is. In the sense, the more often this region is open from any nucleosomes or any other protection. And it would be important, for instance, to search for motifs which are more frequent here, but in a high peak, but less frequent in the low peaks. And that's what we have, developed recently, a tool which is exactly doing that. So you can put in the tool the information about the peak height, and that's in a form like this. So if I open this these peaks as a table, not this one, but this one, I think. So this is these are the peaks. I have this. So you have this property score, which is well, in different software give you different scores, but most often it's correlated with the number of reads which are found for these peaks, which means there are peaks which have very high score, and there are peaks which have very low score. So and then what we do here, we apply the match tool, so the tool which analyzes sites in that areas. So basically what I do, I have run this so-called TRANSFAC match for tracks. I put here those peaks like this, select a profile, which means I select those motifs which I want to find. It can be the full library of profiles from TRANSFAC. And then it searches first for the binding sites inside those peaks. But then there is another tool which is called, it searches for combinations of sites which are correlating with the size of that peak. And in this case, we put here this number of sites which are found. In our case, those sites which were found here. So I put it here, right, like this. And I selected this log score. So I logarithmically convert these scores into logarithmic scale. And then I searches for the combination of binding sites. This is typical CMA, composite module analyst interface. So the genetic algorithm searches for the combinations of binding sites in the windows, which are scaling. And then you run it, and let me show you the results. So it actually found a set of motifs, and among them, again, you see the NANOC and again Fox, which is correlating with this score. But what happens? You will be surprised. Actually, it's in fact anti-correlating. So it's a still quite noisy correlation, but it's quite significant. It's like the p-values are here not shown, but Spearman correlation is minus 0.57 for this number of dots. It's quite significant, and you see like a clear that the higher the score, so which means the more reads you have in the peak, the less is the score of this combination. So which is very interesting, because which means we have seen that such factors as nano and in the Fox and Oct, it's not here, but they in fact are characteristic of the motifs located in the promoter of upregulated genes. But when you start comparing actually here we run it against all ATAC-seq regions in the whole genome, and we found anti-correlation, which means when the peak has a high score, a lot of reads are there. It rather attracts factors to it, which are maybe inhibiting the genes. So at least it's interesting phenomenon. So sometimes quite unexpected things happens, and then this tool allows you to find them. All right, so that's a little glance on the tool which worked to analyze data with information about score. Like it could be a score associated with a with the height of the peak, for instance, like in this case, but it could be other score. For instance, it could be a score which is associated with expression level of the gene. And then this tool will also help you to find motifs which are not only specific for the set of upregulated genes, but also correlating the value of upregulation. So the higher the upregulation, for instance, the more frequency of those combinations of factors you observe there. All right, so that's something I wanted to tell you today. And I don't see any questions so far. Thank you very much for your attention, and we are waiting for you for the next coffee breaks. So we are doing it regularly. Just watch for information on our website, or you will get basically emails reminding you about our events. Thanks a lot, and a goodbye.